Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on chromatography. During the past few lessons in our unit on energy transformation, we learned how plants control light energy in photosynthesis. In the last lesson, we learned about C3 and C4 plants. We discussed the difference between plants and how they photosynthesize. We also looked at a few examples of C3 and C4 plants in Ethiopia. In today's lesson, we are going to look at methods of separating photosynthetic pigments in plants. We will learn about a technique called chromatography. This is a method used to separate photosynthetic pigments as a way to study the substances in more detail. Students, let us begin the lesson with an activity. What is chlorophyll? Take a few minutes to review the definition of chlorophyll with a classmate sitting nearby.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, students. I am sure this was a simple question to answer. Chlorophyll is the green pigment found in almost all plants. It is a very important pigment molecule that enables photosynthesis to occur. Chlorophyll is in fact only one pigment in a group of closely related pigments commonly found in photosynthesizing plants. These pigments are called photosynthetic pigments. Several different classes of pigments are needed in order to maximize the energy taken in from the sun. Chlorophyll is the most prevalent of the pigments, but all are required for the process of photosynthesis to function most efficiently. By utilizing the technique of chromatography, we are able to separate the different pigments of a plant sample. We can see chlorophyll as well as the other pigments that are in the plant. There are actually quite a few different types of chromatography techniques, including paper chromatography, thin layer chromatography, ion concentration chromatography, and gas-liquid chromatography. Chromatography consists of two phases, the stationary phase and the mobile phase. The stationary phase is comprised of a stationary item that allows the pigment molecules to pass through it at different times based on their size. The mobile phase is the solvent that dissolves the substance being tested. Before we discuss the process of using paper chromatography, let us make sure we understand the role of pigments in photosynthesis. Can you explain what happens during photosynthesis when pigment molecules absorb light energy from the sun?
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, students. I hope you were able to remember your studies on photosynthesis. Once pigment molecules absorb light energy, it is converted into chemical energy. The chemical energy is transferred and used to produce glucose molecules that are vital to plant development. It is important for plants to have a variety of pigments. The more pigments it has, the better it can utilize different wavelengths. Several pigments can capture more light energy than just one pigment. Let us continue on with the lesson. Paper chromatography uses sheets of paper as the absorbent stationary phase. Acetone is commonly used for the mobile phase. A concentrated spot of leaf extract is applied to the paper. When the paper is dipped into the solvent, the pigments move up the paper at different rates because they have different solubility. The pigments become separated from one another and can be identified by their different colors and positions on the paper. The more soluble a pigment is, the greater distance it will travel on the paper. If the pigment strongly absorbs into the paper, the slower it will travel up the paper. With luck, you should be able to identify 5 pigments: chlorophyll A, which is blue-green, chlorophyll B is yellow-green, xanthophyll is yellow, carotene is orange, and pheophytin is gray. If you refer to Activity 5.10 in your textbook, you can see the directions on how to conduct a paper chromatography experiment using a spinach leaf. Spinach leaves are perfect examples to use for this experiment. They contain chlorophyll as well as other pigments which are necessary in photosynthesis. Let us review what we have learned, students. Please define the process of chromatography.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello, students. Chromatography is the technique used to separate chemically similar substances in a mixture as a way to study the substances in more detail. Is this similar to your answer? Great work, everyone. Let us do one more activity. Students, explain why different pigments in leaves are beneficial in photosynthesis.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello once again. A spectrum of pigments is beneficial because more of the wavelengths of light can be utilized. By having a variety of pigments, the leaf is capable of absorbing as much light energy as possible. This enables efficient photosynthesis in the plant. This brings us to the end of our lesson. Today, we learned about the technique of chromatography in demonstrating the photosynthetic pigments within a plant. Until next time, thank you, teacher. Thank you, students.